So as always, when we do these briefings, we set the stage. We have expert people provide wonderful insights and set the stage on some of the information. And then we could spend the rest of the day talking about all the issues that have been raised. And sadly, we only have about 25 minutes, 20 minutes left in our briefing time. So most of all, I'd certainly like to make sure that you all have the opportunity to ask questions. And I'm never at a loss for words, so I'm certainly keep some conversation moving along. But again, I'm looking to all of you to raise questions, and I'll call on you now or at any point that you raise your hand. So I'll start first by just opening up the floor. Yes.
Hi, Erica Siski, Senator Franken's office. I was wondering, I appreciated Mr. Lazarus' point about, I think it was you, about reimbursement. I think that's a piece that's often left out of the conversation, and ultimately, in order to really achieve, the goal of personalized medicine, people have to have access to it. And we just heard from a consumer perspective that lack of payment means, it's sort of a moot point. So I was actually wondering if the FDA or NIH folks could comment on how this new working group with CMS might help sort of the translation of approval and sort of affirming validity to payment, or if we start looking at alternative frameworks for approving these kinds of diagnostics, how do we have kind of a minimum threshold so that payers feel some kind of confidence that it's something that ought to be a covered service?
Great, thanks. Who wants to catch that one first?
Sally, you want
to? Of course, CMS isn't actually here, so you realize you're.
Sorry, yeah, they're not here.
So FDA has a working group with CMS around the laboratory developed test framework. And that working group was actually designed mainly to map out the differences between CLIA and FDA. So, but I will say, one of the things in the Precision Medicine Initiative has been to touch on the relevance of reimbursement and the need to address that. In a different setting, we haven't done this in the precision medicine setting yet, but in a different setting, CDRH, and that's our Center for Drugs and Diagnostics, or for diagnostics, and that covers the devices and diagnostics. They've got a parallel review that they've done with CMS before, and so, and it recognizes this. It recognizes that we have a standard that you have to meet in order to get your test approved. And CMS has a standard that you have to meet in order to get your diagnostic approved for reimbursement. And we should be able to, to marry that up as much as we can to see if, as you're going through one pathway, you're also sort of getting, at least checking some boxes for the other pathway. There's a lot of work that needs to be done still on that.
It just seems like, to the point about why it wouldn't be practical for the FDA to be sort of reviewing every sort of mutation. And so, obviously LDTs and future diagnostics are very complex. So, obviously didn't mean to put you on the spot. Just wanted to know kind of what the was.
Kathy, I don't know if you, any particular view from an NIH perspective on reimbursement?
No, I really don't. I don't want to speak for CMS, but I do think that there's an important observation that can be made here. So there is a very large number of molecular diagnostics that are out there available as laboratory-developed tests where there has not been a third party who has looked very carefully about the analytical and clinical validity of those tests. And to the extent that FDA is moving in to flexibly and creatively be able to oversee and validate the quality, the analytic validity and the clinical validity of those tests, that gives CMS something to work from rather than just take our word for it. If FDA is weighing in, there's an additional third-party validation that I think is going to be important for CMS.
Yeah.
I just wanted to mention that one of the most difficult things is the transition from a test, especially tests that have multiple genomic targets, going from the research, clinical research environment or the translation medicine environment and then going to be submitted to the agency and becoming a day-to-day clinical workhorse. So obviously, when you're in the research and translational area, you want to cast a wide net. But when you are deploying a test for day-to-day clinical use, you want to have a very focused group of clinically efficacious, proven targets. And it's very hard for people to give up the wide net. That's why, for me, ultimately, working with FDA is really is such an important thing, and I'm so happy that we do that, and we obviously, as an IVD manufacturer, we're not on the LDT side because it forces us to have this dialogue and to be very focused on deployment for the use of caregiving pathologists, caregiving laboratories, and caregiving oncologists, et cetera. It's a very different mission when you have that kind of test as compared to the way you make a test when you're casting a wide net.
Nancy, you wanted to weigh in?
Yeah, just to add to what Kathy said, I talked to a lot of community oncologists and people, patients out in the community. And one of the things I've heard from the oncologists is they don't know what to do when a patient comes in, when a patient is paid to have their tumor analyzed by a third party like Foundation Med or Caris, and they come in with this huge list of things, and the doc is like, well, I don't know what to do with it. We don't have any data to say you should do this or you should do that. And I'm glad to see FDA moving in that direction because that's a really confusing area and it's not reimbursed and people are paying. I mean, I've talked to people who've put second mortgages on their house, which that shouldn't happen.
Academic standpoint, the clinical standpoint, there's really two types of cancers right now. There are cancers where we can bill for testing, and then there are cancers where we cannot bill for testing. There are only 7 adult human cancers that we believe we can bill for. Those include lung cancer, melanoma, colon cancer, certain types of sarcoma, and a few other rare subtypes. The way next-gen sequencing works is that once you have a cancer where you can bill for next-gen sequencing, there's almost zero additional marginal cost to doing 400 genes versus doing 50 genes. So once the cancer is billable, those cancers are going to get sequenced, and what's going to happen is we're going to not be doing a few genes soon. We're going to be doing hundreds or potentially all the genes and charging the same amount of money as we were charging for a small gene panel. The challenge right now is for the non-billable cancers, which include some very common cancer types like prostate and breast, ovarian, endometrial, kidney cancer. For these type of cancers, you cannot bill, at least in our opinion, for any genetic testing in those cancers. And so I think it is problematic when patients have this type of testing and they're not plugged into an infrastructure where they can be put on experimental clinical trials, which is really the main reason to do that type of testing.
So I want to offer up just a quick observation on kind of a framework for thinking about these things that may be useful, particularly for those of you who are newer to this area. And that is to realize that from both a practical standpoint and from a policy standpoint, you should be thinking always about 3 dimensions of diagnostics. And the first, which some of our speakers have touched on, is analytical validity, which basically means how accurately and precisely does the test measure the thing it's looking for. And test manufacturers, test developers will specify the degree of precision and so forth that their test offers. And that's relatively straightforward, not always, but that's, again, the sort of the threshold for a test performance. The second dimension of diagnostics is clinical validity, which also our speakers have touched on, and that's really the measurement of whether the test result is relevant to the disease state or condition that you're looking at. In other words, is that test result going to tell you something meaningful about that disease state? That's FDA's second approval point. First on analytical validity, and then secondly on is this result actually clinically valid? Can you make claims about how useful this result is for something in particular? The third dimension, which is what I particularly wanted to get to because it's so relevant to the discussion around reimbursement that everyone's always interested in, and everybody, no matter their area here, always talks about reimbursement. That's clinical utility, right? So the clinical utility of a test is a measure of how much having that result and following that result when you're making a clinical decision changes quality of care somewhere along the line, or it changes the ultimate outcome. To what extent does having that particular result change care, as opposed to doing something else or not having that result? That's a very complex field that is not in any way very well resolved, and there are a myriad of private payers out there, in addition to, of course, we talk a lot about CMS with Medicare, but there's a great disparity among payers in the kind of models they use to determine clinical utility. And it's a field that in diagnostics is nowhere near as well developed and studied and thought about and opined about as it is in pharmaceuticals, for example. And so this leads right now to a lot of this kind of churn in the system and delay and sand in the gears around moving more quickly to having private payers and Medicare cover tests that maybe a broad swath of people think they should be covering to provide a pull-through of important technologies. And so to just land quickly on that, I think that it's one of the potentially the ultimate benefits of initiatives like the Precision Medicine Initiative is to come up with new ways of aggregating data, new ways of more efficiently looking at large amounts of data. And we've talked a little bit about shifting models of clinical trials. All of these things are ways of starting to get a handle on the tremendous amount of information that we now can glean, and potentially moving more quickly to a point where payers are more willing to pay for tests more quickly and therefore drive them into wider adoption. So I just wanted to very quickly set that stage. I think it's a useful way to kind of think about all these issues we're wrestling with. So I don't want to hog the floor, obviously. Other questions from the audience? Yeah.
An issue about educating the public and educating physicians so that they know which tests validate, which tests are used, which patients have them, and the patient should be asking. This is something that we're going to try to work on more. I'm sure you are as well. So there's a whole sort of vacuum in terms of people, outside of where we are now, knowing about these diagnostics and precision medicine. Need to know about them in order to use them.
Yeah. I don't know, Nancy, do you have a comment on that challenge?
Yeah, I talk about getting a diagnosis of cancer is like being dropped out of an airplane into a foreign country with no map, no translator, and you need to go someplace, and you have no clue how to do it. The amount of disconnect in your head at that moment of time is just overwhelming. And so I think to expect the public, to expect patients to figure this out is going to be challenging. I think educating healthcare providers is really the really critical piece. And to the point about pathologists, not every oncologist has I'd love for everybody to get treated at Sloan Kettering. Where I come from, there isn't a pathologist. You call the one in the big city, and the guy in the big city calls back and says, oh, this is what it is.
Dr. Sullivan, I think you might have a helpful perspective on some of this. And I think we tend to look at the latest results of trials or hear about the initiatives that you're running, and we think, oh, we're on the verge of having everybody in the country and having every community cancer doc have access to and be able to use the kind of information that's emerging. But obviously, that's not the case. And would you care to comment on the distance between what you do and getting things into standard of care that you were referencing earlier?
I really think that there's going to have to be a major push for physician education in oncology, because this is going to happen, and it's going to happen very quickly. So I do think there's a tipping point. If right now 30% of patients have tumors that should be genetically tested already as standard of care, that's probably going to shift to 70, 80% just in the next 2 years. And that means once it shifts to that frequency of testing as standard of care, I think it'll just, result in everyone will be tested. So there's going to have to be physician education. I think you have to recall that many physicians went through medical school and none of this was ever mentioned. So this is the classic need for continuing medical education. And it's not that they weren't listening in medical school. None of this technology or none of this knowledge even existed. I also think we have to, I actually think the testing is actually going to be fairly easy to overcome this issue. Once, again, every cancer becomes billable for testing, then it's really going to be a commodity. And its question is who can offer the cheapest and most comprehensive testing with the highest quality at the lowest price. And I think that that's going to be where we are in a few years, and that issue will be solved. The bigger issue is what you do with the information when you have thousands of data points because thousands of genes were tested at the same time. Again, there it's physician education. I think we can use computer software in terms of learning tools to sort of remind physicians, okay, the patient has an L858R EGFR mutation, that means they should be getting erlotinib. And so it's something that computer tools could really help remind the physician what they're supposed to be doing, because there's hundreds or there's going to be thousands of those specific mutation drug associations, and nobody can easily remember them all. So why not have a database that automatically comes up when the mutation comes up to remind people what they're supposed to be giving and what's the evidence for that? And then I think we also have to change the way we do clinical trials. I mentioned to you that basket study where that's a pretty broadly available clinical trial. It's going to be open at about 30 centers, but that means probably the majority of patients in the country don't live within driving distance of a site where that clinical trial is being offered. So instead of having patients come to places like Sloan Kettering for these clinical trials, is there some way to bring these clinical trials to the patient? And that's, I think, somewhere where the NCI could really make a big impact, because you could have these clinical trials based on a national level through a central IRB, which would approve the clinical trial, and then maybe that clinical trial only opens at that local site when the patient is actually found who needs that clinical trial. So when the genetic result comes across, there would be some computer learning tool that would say, okay, the perfect clinical trial for this patient is one that's open through this central IRB. It only is going to take 24 hours to open that clinical trial, and that could be done right away. So I think we could even develop new ways to do those studies, because not everyone can travel from Oregon to New York City, and I'm not sure you would want to, even if you could. So.
Question back here.
I'm Steve Pearson with the American Statistical Association, and this is not a statistical question, but I think it's for Dr. Stolen. For just kind of the scope of what we're aiming for, you showed the study of bladder cancer where 145 patients seem to respond to this. So, given that there's many different kinds of cancers and potentially 7 billion different individuals, how many different treatments or drugs would it take to cure 75% of all bladder cancers, or 90%, or?
So I think that there's two responses I would give to that. One is that there really are drivers of patients' cancers that are only found in a very tiny number of patients. And simply because those things are rare doesn't mean they're not important, because they're important for each of those individuals. And in many cases, we already have a drug that works against that drug target that we can use. And I think a good example would be a very rare cancer called histiocytosis that was studied in one of these baskets. There's only about 500 patients a year in the country at any one time that have this cancer. And actually was a drug that Nancy mentioned called a BRAF inhibitor. Turns out that half of those patients have BRAF mutations. 100% of those patients respond to the BRAF inhibitor when they're given the drug, but you'll never be able to do randomized studies because there's so few of these individuals in the country who have active disease at any one time. And so you're going to have to change the model that we're going to be able to develop drugs in these rare instances. And that's one that was easy because the BRAF inhibitor was already developed in melanoma, but there are other instances where there might be really orphan targets that are only going to be relevant in a very tiny number of people, and can you come up with a model to develop a drug there? So that's one issue. To get the second part of your question, how many drugs do we need to develop effective therapies for all the patients with bladder cancer, as an example? There is no real answer to that question, and it's not because we don't know the genetics yet. We actually, the Tumor Cancer and Genome Atlas has now defined the genetics of bladder cancer to some degree. But in about 70% of patients with bladder cancer, the genetic alteration that drives their tumor to grow, there is no drug yet available against that genetic alteration. So this is, again, one of the downsides to testing, is that you may identify a mutation for which we don't have a therapy. Now, that doesn't mean mutation testing was not useful for those patients, because it showed that all these other treatments were not going to work. So don't waste your time with those, and maybe try something that is mutation independent, like immunotherapy, or maybe an anti-angiogenic therapy, or some other stromally based target. So that's a bigger challenge as well. But right now, I would say for bladder cancers, it's only a minority of patients for which we've got good targets, but you still have to test everyone to find that minority. For other cancers like melanoma, most of the patients have an interesting drug target, and that's probably why melanoma testing is now standard of care.
So we have by no means exhausted this topic, and as I often say, we have exhausted our briefing time, however, and I just wanted to come back to you, Kathy, and ask you one quick question very specifically on the use of next-gen sequencing in the million-person cohort that you're planning to develop. How widely do you expect at this point that NGS would be used to perform sequencing on those individuals? How many individuals? And are you talking about a full genome sequence or just some targets that you will identify?
So the short answer is we don't know yet. I'm going to guess that we're going to start out with some informative genotyping and sort of watch as the cost drops for whole exome and whole genome sequencing and add that a little bit later. But we would definitely start with some good genotyping at the get-go, especially for markers that are informative about drugs and drug response.
Great, thank you. Well, I hope that we have provided yet more information for all of you and whetted the appetite of those who are new to this field. I want to ask John Retzlaff, who's Managing Director of Science Policy and Government Affairs with AACR, to offer some concluding remarks, and I want to again thank AACR for their collaboration on this.
Yeah, well, thanks so much for everybody coming today. I think it's been an excellent briefing in many ways. AACR has had an opportunity, now it's a second time, to work with AdvaMed DX. About a year and a half ago, we had a conference about the importance of diagnostics in cancer care. And then today, you saw how diagnostics is going to help really advance precision medicine. But I especially want to thank really the leaders. We have two influential leaders from the two agencies who will be leading the whole effort on precision medicine. So Dr. Hudson and Ms. Howard, it's been wonderful to have NIH and FDA represented here at this briefing today. And then we've also, I think it's been very beneficial to see the entire really medical research ecosystem here today. When you talk about discovery and the research that's going on through Dr. Solit's work, as well as the development of these diagnostic tools that are so important going forward, Dr. Lazarus, and finally, most importantly, the delivery through Nancy Roach's work with colorectal cancer and trying to get this progress to patients going forward. So I guess our final remark would be, we hope to go forward and see a very robust and sustained increase for NIH and FDA funding in the FY 2016 budget, which would hopefully include that $215 million that the President has proposed for the Precision Medicine Initiative. So once again, thank you all for coming, and thank to the speakers.